vegan diets without proper supplementation are epigenetically incomplete.
How does epigenetics really affect nutrition?
Diet is one of the most powerful exposome factors influencing our epigenomes. There are some nutrients in the food we eat, epi-nutrients, that are really essential for the proper functioning of the epigenome.
Look at someone's epigenetic or genetic profile and say, this is what you should eat and this is why.
First of all, let me take a step back because...
Welcome to the Everything Epigenetics podcast, where we discuss DNA regulation and the insights it can tell you about your health. I'm Hanna Went, and I'm the founder of Everything Epigenetics. Today, my guest is Dr. Lucia Aronica. Today, we're going to be really diving deep into epigenetics and nutrition. Lucia is an expert in this space. More specifically, we're going to be covering a basic epigenetic overview. What does this really mean? How does this relate to nutrition in the first place? We'll also be talking about epinutrients and examples of those and how we can actually use epinutrients to lower, hopefully, our biological age. On other hands, you can increase it as well if you're not getting enough, maybe, of those epinutrients. We'll talk about sulforaphane, which is a powerful epinutrient. And we'll also talk about nutrigenetics. So are you able to create a diet plan based off of your genetic makeup? We'll talk about two of her most popular studies, the DIETFITS Study, which is looking at low-carb versus low-fat diet as it relates to genetics, epigenetics, metabolic health, and other factors. And then we end with a review of the twin study, where they look at an omnivore versus vegan diet, and they look at epigenetic clocks, again, genetic makeup, some further information included in the study too. Without further ado, a quick bio for my guest. Dr. Lucia Aronica is a lecturer at Stanford University. She is really well known for the genomics R&D lead at Nutrigenetics and is an acclaimed speaker in the field of personalized lifestyle medicine. Her research and teaching really focuses on that nutrigenomic component I mentioned, which is the science of how nutrition, genetics, and epigenetics interact with one another to impact our health and longevity across our lifetime. Dr. Aronica established the first certificate program in nutrigenomics at Stanford and has helped thousands of professionals implement nutrigenomics in their business. And without further ado, here is my guest, Lucia Aronica. Everything Epigenetics podcast, Lucia. I am super excited to have you today. Thank you so much for joining me.
Thank you very much for having me, Anna. It's a pleasure.
Yeah, I'm super excited to chat with you and get back into recording my podcast. I believe you'll be the very first episode of season 2, and I'll release this here soon. And I really want to explore your expertise, which is epigenetics, of course, what we're here to talk about today, and really nutrition, the real science behind how our DNA and diet dance together. I think you put beautifully sometimes to really determine our metabolic health, whether it be right now or later on in life. So I really want to start with the basics here, re-engaging and reintroducing my audience. How does epigenetics really affect nutrition? And maybe I'm even jumping the gun because I'm so excited to dive into it. Feel free to even give us a little bit of your background and how you got started in nutrition, ending up with how nutrition really affects our epigenetic expression.
Yes, perhaps I will start with a very short background about me. I'm from Italy, of course, from my accent, you can realize. And I was born in Italy and did my master's studies there. And then I've been in the epigenetic field for over 17 years, researching first I started with the small RNAs, a type of epigenetic modification or molecule, and then moving to histone modifications, and finally to DNA methylation, which is a type of epigenetic modification that we can reliably measure with accuracy at a single nucleotide level. I'm talking about that because later on we will talk about tests like epigenetic clocks that are based on DNA methylation. And I'm studying now DNA methylation in the context of nutrition and specifically clinical trials that compare different types of diets. Like most of our studies focus on low carb versus low fat diets. And one of our latest study that was also featured in a Netflix documentary, You Are What You Eat, a twin experiment, was looking at the comparison between a vegan versus an omnivorous diet. And we will talk about all these later. But now let's start from the basics. I'm sure your audience is familiar with epigenetics, but just to sum up, epigenetics from the Greek prefix epi, meaning on the top, describes a layer of molecular instructions like these methyl groups, but there are many other molecules that regulate gene expression, modulating genes up and down. Now, unlike our genes, which don't change during time, over our lifetime, epigenetic modifications are dynamically modified by lifestyle factors such as diet, exercise, but also stress, sleep, environmental toxicants. And we call all these lifestyle factors and our lifetime exposure to them the exposome. Now, coming to your question, diet is one of the most powerful exposome factors influencing our epigenomes. And there are many studies showing this in animals and in human beings. Specifically, we know that there are some nutrients in the food we eat that I call epi-nutrients that that are really essential for the proper functioning of the epigenome. Some of these nutrients are primarily found in animal foods, specifically so-called methyl donors, which donate these methyl groups that are essential for DNA methylation and other epigenetic modifications, but also for several other biological processes beyond epigenetics, like DNA synthesis of crucial molecules like creatine, phosphatidylcholine. So basically, methylation is at the center of our physical and mental health. And the second category of epinutrients comprise a set of phytochemicals, so molecules that are found primarily in plant foods that can modulate the activity of enzymes that can write or erase epigenetic modifications from our genes, thus affecting gene expression. And all these phytochemicals comprise things like polyphenols, like flavonoids, for example. My favorite ones are the one found in olive oils, chocolate, dark chocolate, coffee. But popular ones also include curcumin, the active ingredient of turmeric, and also anthocyanins, like there are polyphenols believed to be very beneficial for brain health, found, for example, in things like blueberries. Also, we have carotenoids that have been associated always with the younger epigenetic age. We can talk about that later. And we also have a very interesting class of these epi-bioactives, how I call the second category of phytochemicals that are basically produced when we cut or chew or chop some plant foods like garlic or broccoli and cruciferous vegetables. So they are not contained in the plant itself. But when we chop and eat these plants, we activate a reaction whereby these compounds are produced. And one of the most famous examples is sulforaphane in broccoli and cruciferous vegetables, which I think the magic about sulforaphane is it's that it isn't itself an antioxidant, but it can turn on a cascade of antioxidant and anti-inflammatory genes. So actually it's even more powerful than something like vitamin C, that you take it and only lasts 2 hours, whereas sulforaphane, you take it and in experiments, the gene regulating effects are lasting 72 hours. So maybe you can eat like a portion of broccoli every 2-3 days and enjoy the epigenetic benefits of turning on your own antioxidant defense system. So this is what I call epinutrition. Is just understanding the fundamental nutrients and building blocks. Of course, not only what we eat influences our epigenome, but also when we eat. We know that, for example, fasting, intermittent fasting can also influence our epigenome, and sometimes even the macronutrient composition. So we know, for example, that on a ketogenic diet, we produce ketone bodies, and ketone bodies have actually an epigenetic function. They are produced on both fasting and the ketogenic diet, and they have several epigenetic functions that are primarily anti-inflammatory, but we still don't know all the functions. We know that ketogenic diets are therapeutically used for a variety of conditions, especially traditionally, like epileptic children that are resistant to drug therapy, but also now mental health conditions. So, and it's possible that the epigenetic effects of ketones contribute to these benefits.
Yeah. That's lovely, Lucia. Thank you for laying that groundwork. I appreciate that nice answer. And one point that always comes up too when I'm speaking with healthcare providers about these epigenetic clocks and really using them is we always now hear this term. I think Dr. Mark Hyman uses it as well. You are what you eat, or food is medicine, right? You see it all over social media. It's almost like it's explaining or talking about epigenetics without really directly saying it. So that's what I think you just explained, is really going deep into those epi-nutrients. And I gotta say, I would probably agree with you there. I'm a huge fan of olive oil, chocolate, coffee. I probably could have, pick a favorite out of each of those. But yeah, those really containing these epi-nutrients to modulate epigenetic expression in ways that are beneficial for our biology is really key in understanding, maybe which ones are right for you. Now, one thing as well, Lucia, I remember listening to you speak at PLMI this past year, Dr. Jeffrey Bland's conference, and I loved your slides. I was picturing them in my mind as you were saying, if you have adding and you're taking away these methyl groups, you're turning things on and off, and it's like the pencil eraser. I think you had a really nice slide that actually showed almost like an animation of writing and erasing, which I think is a really good comparison as well. And the last thing I'm going to say, and then ask you some more questions, is I've recently been going down this own journey myself with sulforaphane. As a supplement that I'm taking. So there's a really, really cool company. Of course, you can eat the broccoli as well and want to make sure you're getting enough naturally through foods. But if you're still a little bit lower and you're like me, where I know I have a lot of oxidative stress build up and reactive oxygen species in my body, I do this I really like this company called Brockshot. It's a really kind of natural, all-encompassing company that is going to make sure you're getting those exact sprouts you need to form sulforaphane that's going to convert correctly into your body. So I wasn't sure if you were aware or if there's any other, supplement-based sulforaphane companies out there that you like as well.
That's very interesting. So I'm not familiar with this company. I know that there are many companies, and basically what you find on the market, as I mentioned, sulforaphane is produced from a precursor that is called glucoraphanin. And when you cut or chop broccoli, then you basically release myrosinase, the enzyme that acts on glucoraphanin to produce sulforaphane. While I'm explaining this to you, because this is important to understand the different supplements you find on the market. Some of them only have glucoraphanin, and unfortunately, these don't work because basically, I think it's only 1% that you convert on average. I don't remember the exact percentage, so it's very little. It depends on your microbiome. The one that that contain myrosinase, glucoraphanin plus myrosinase, they are better, but they're still not elevating your sulforaphane level. We don't know why. Probably because myrosinase is destroyed in the gut. And then we have the third category. Maybe this is what your supplement is about, where you have stabilized sulforaphane, which is patented. I think only some companies do it. I'm familiar with one. I think the brand name is Sulfodyne, and is used for prostate cancer. Is Prostane the product? I don't remember but it's actually is a company based in Europe.
Interesting.
Yeah,
I'll send it to you afterwards. I know I always forget the names of the precursors. It contains the one that starts with a G and the myrosinase, so it contains both of them. Okay,
so it's good, but probably the best would be stabilized sulforaphane.
Stabilize it. Yeah, they may stabilize it too. I don't know. I'll send it to you afterwards.
So it could the best would be taking actually the sulforaphane, the end product, not the precursor. That's that this is what has been studied used in many studies. So the stabilized sulforaphane, which is I think more expensive, but I agree. I mean, supplementation sometimes can be a healthy shortcut to.
I think people need to understand just what to look for, right? And kind of the differences, which I think is really, really important to talk about. So I'll shoot you an email afterwards and send it to you. But what I want to talk about now, Lucia, is, how does one's individual, let's say even genetic, we can talk about epigenetic makeup too, but how does that really influence which diet is right for them? Can you look at someone's epigenetic or genetic profile and say, this is what you should eat, and this is why, this is the science behind it? Are we really there yet?
Okay, so first of all, let me take a step back, because what we talked about is called epigenetics. So how actually the food we eat can affect our genes, gene expression through epigenetics. Now, the flip side of that is called nutrigenetics, what you are asking me now about. So how our genes can affect our response to nutrients. And nutriepigenetics and nutrigenetics work hand in hand in nutrigenomics, which is basically an umbrella term to describe everything about the interaction between genes and nutrition. And so it's nutrigenetics and nutriepigenetics. Now, of course, yes, this is a dialogue. So the I can only say yes, of course, genes do affect many aspects on our response to food, from obvious examples like food intolerances, lactose intolerance is a famous example, to even our preference for sweet foods versus salty foods. We know there are some genes influencing that, and people who have a predisposition for a sweet tooth, they also have actually a double rate of caries and problems and dental problems. So yes, there is an influence. Now, I think your second question is, are we there yet? Can we already make recommendation based on your genetics? And I would say it depends. There are things where, yes, there are some genetic variants for nutrigenetic variants that are backed by science. And then there are, for example, you may have heard about MTHFR, is a gene that basically produces an enzyme, MTHFR, that activates folate or vitamin B9 from the food we eat. So if we have a variant, a very common variant in this gene, so this is not a mutation. Many people call this a mutation. A mutation is a very rare genetic variant, and it's like 1 in 100,000 or million people, and it's rare for a reason, because it's not been selected by evolution. And so very often is associated with disease. So when doctors say that you have a mutation in MTHFR, patients are scared, and rightly so. And this is because it is a mistake it's not a mutation, it's called a single nucleotide polymorphisms. There are many variations, and it's common. For example, the region where I was born, Campania, Italy, there are actually more people carrying these polymorphisms, like 60%. 30% have two copies. My mom is one. My mom she's 83 years old, vibrant. She has 4 kids. And, and she has 2 copies of this polymorphism that presumably predisposes you to have like miscarriage and all sorts of diseases. And so, of course, this is a predisposition, a weakness, but that needs to be expressed, that only expresses itself in a context. So we were talking about nutrigenetics. This is an example of that. It can be useful, however, to know that we are a carrier, as many people, or we have 2 copies, just because in this way we can make sure to actually do what we are supposed to do anyway. So, which is eat a diet that is rich enough in folate to provide more opportunities for this enzyme to actually activate the enzyme. And folate is found, like, you can eat the folate you need in one day, just with less than 100 calories from 200 grams of spinach or 30 grams of liver. And then we also need more choline if we carry this polymorphism, because choline works with folate in its cellular functions, which are detoxifying homocysteine and powering methylation reactions. So choline is found primarily in eggs and liver, a little bit in meat, and then only in limited amounts in plant foods like broccoli and nuts. So, knowing this, it can actually motivate us to meet our dietary requirements. So I think there are some variants like this one, and there are many other ones that can predict how many, if you can activate the omega-3s from plants food to the active one we need, like EPA and DHA. These variants, I think they are useful to motivate people to actually do what they are anyway supposed to do. Even with
Yeah.
vitamin D, there are things that we should do, but we can motivate ourselves. And this is called also the nudge theory. So actually, I think Richard Thaler won the Nobel Prize in economics recently, talks about this theory whereby little nudges like this one, like indirect suggestions, like you have these genes, you maybe should be doing that, are actually more powerful than prescriptions. Eat your spinach, eat your choline. It's so I think this is where the value of nutrigenetics and lies, because in the end the recommendations are basically we should be doing things that we should, nothing really, but and then there are things where I think we are not there yet, and probably we will never be. For example, recommending a low carb or low fat diet or keto diet based on genetics. And unfortunately, there are companies that claim that this is working. We have studies showing, some studies show that, but the studies were not done with truly low carb diet or even keto diet.
Got it.
We wrote a review that I can link if you want. I can send you the
Please.
article. Yes, where we actually talk about this problem. So that there are no studies on keto diet and very low carb diet with nutrigenetics, and yet you have company saying keto is not for you, or low carb. So it's crazy. And in one of our study, the DIETFITS study, the Stanford DIETFITS Study, this was the largest randomized clinical trial ever undertaken to compare a low carb versus a low fat diet with 609 people enrolled, overweight. Anyway, we tested 3 of these SNPs that are commonly included in this DNA diet, claiming that they can predict whether you are going to respond to lose more weight in response to a low carb or low fat diet. We didn't find any signal whatsoever. And our diet was more like a truly low carb diet, especially in the first months. With less, so people in the first months were eating less than 20-30 grams for carbohydrate, and then they increased like 100 up to 100 grams at 12 months, but was still low carb, truly, a low carb diet. So I think there is a good there is a possible good use of nutrigenetics, and the more reputable companies can provide those services.
It's hard, I think to sum that up too. Thank you for that answer and the transparency and the honesty. So sure, based on a couple SNPs or variations, I like that you used genetic variation instead of mutation. I know we're trying to move away from that because there is a big difference there, especially when you're talking one-on-one with a patient. So maybe there are a couple variations of that gene, that SNP, that single nucleotide polymorphism, that could give you hints or suggestions on what you should focus on. But we're not saying, here's your genetic makeup. This is what you need to eat for breakfast, lunch, and dinner to reach X goal, right?
Yeah. And if I can add something here, because I think the second problem is that also, let's say that it's true that our genetics, for example, can influence the way we respond to carbohydrates. So we may well have some genes that predispose us that protect us from developing diabetes, that increase our tolerance to carbohydrates. But let's say that you have been eating all your life refined carbohydrates and sugar. Very likely, by the time you are 40, you have some either pre-diabetes or some glucose tolerance problem independently of your genes. So getting a test that tells you, go go more carbs and more, that it may actually be detrimental. So
Interesting.
there is a predisposition, and there are other metabolic factors and markers that must be integrated. For a truly personalized plan. You should have nutrigenetics and then integrate that with other markers and see whether they can confirm that predisposition. Even with vitamin D, if you are predisposed to be deficient, but then you are doing all the right things not to be deficient, then you don't need to take more, right? So
Absolutely.
yeah, so always have combine different tests.
And I'm an advocate for that as well. And you also need, to test. If you're doing any type of repeat-based testing, you need a baseline. You need to work in all of those results together. We're getting more and more toward this personalized journey and healthcare. It's really, truly N of one precision-based medicine. And sometimes I can get a little frustrated when everyone's like, what should I take? What diet should I be on? What should I do? And I can give them very general answers based on these large populational based studies, but they still need to do that, N of one testing on themselves to really understand what works for their body and their biology. So, that makes perfect sense. And, it leads perfectly into my next point. I was going to bring up that DIETFITS study that you mentioned. You already kind of gave us a really great background. It's, like you mentioned, the largest study ever undertaken in personalized nutrition, looking at low carb versus low fat diets. And that, I think, primary goal of the study, you can correct me if I'm wrong, was really to understand how weight loss affects gene activity through these epigenetic modifications, and whether we can actually use those modifications to predict diet response for a personalized weight loss strategy or just in general. And you said, no, there's really not that strong signal there. Is there anything else that you want to add about that DIETFITS study before we go into the twin study?
Yes, is that, as you say, this is one of the largest study in personalized nutrition, because we basically, the rationale of the study is that one diet doesn't fit all. So we collected lots of molecular markers, trying to identify those markers that could predict who is going to respond best to which diet. So genetics didn't at least show the three genes that we tested didn't seem to predict that. And then we also included many other markers, including epigenetics, but also microbiome, metabolome. And I was leading the epigenetic analysis. So what we found, we didn't find epigenetic markers that could predict who was going to respond best, but we found interesting things. So the first thing we found, and we didn't publish the results yet, we had so many other things to publish and work on, but we will publish that eventually. So one thing is that we saw that some epigenetic markers of diabetes, like a marker that you also include in your test, ABCG1, basically showed an improvement on both diets after weight loss, basically showing what we were talking about before, that epigenetics is modifiable by nutrition and weight loss. And so if basically what happens is that when we gain weight, there is this particular genes, and this is just one example, that gets methylated and turned off. And this is a gene involved in diabetes. And then when we lose weight, the good news is that some of these epigenetic marks are written in pencil, so we can erase them. We have the power to influence our epigenetics, and this is associated with health outcomes. In this case, this is an important gene in diabetes, and we can turn the genes back on again. So this was a very exciting finding. And then we also found that there were clear difference in gene expression between people on a low carb and a low fat diet. And interestingly, for example, the people on a low carb diet upregulated their fat metabolizing enzymes. So and this makes sense. If you eat more fat, because on a low carbohydrate diet, you eat more fat, you are going basically to turn on the genes that make you break down fat. And this also brings us back on the conversation on nutrigenetics and nutrigenetics, because let's say that, genetically you don't have good fat metabolizing genes, but then you go on a diet and you turn those genes on. So there is always like, yes, the predisposition, but then is the lifestyle that also contributes to the efficiency of those enzymes. So it's always a conversation. So I think these were the most interesting findings for that analysis.
Yeah, and it really is that nature versus nurture pool, where
Yeah.
your nature is going to be that nutrigenomics, if you will, or your genetics, and the nurture is going to be the epi-nutrients, if you will, or the epigenetics. And I think we're still trying to figure out, I think we are learning that possibly epigenetics makes up, a bigger chunk of these health outcomes, or has a larger relationship to all-cause mortality and morbidity compared to genetics. I think we're still trying to figure out, is it 80-20? Is it, 60-40, or where that break really is? But the power is that it's all in your own hands now, right? You're in the driver's seat. You can understand your genetic predispositions, but there are a lot of positions where we can start to, again, add these methyl groups and shut genes off that we don't want to turn on, or take away these methyl groups, express genes that we do want turned on. And you mentioned that ABCG1 gene as a very specific example that's related to type 2 diabetes. So I think, even the reason behind us having this conversation is to make people aware about this. Your health is in your own hands. We can make even some of those intuitive lifestyle choices that may relate to diet, right? Instead of grabbing for the candy bar right before you're going to go to bed, either skip it, or again, smaller portions of that compared to veggie fruit intake. So there's that intuitive piece in there as well, but you can really start to get more personalized once you're looking at that individual level. So what I want to go over now, Lucia, is we're getting to the end here, maybe about 10 more minutes left or so. I want to spend the rest of the time talking about the twins study on Netflix that drew a lot of commentary and eyes. It's the You Are What You Eat documentary series, and this is based really on a particular study we did together called Unveiling the Epigenetic Impact of Vegan versus Omnivore Diets on Aging, Insights from the Twins Nutritional Study. And of course, that's going to look at those different effects of vegan versus omnivore diets on epigenetic aging and a lot of other related biomarkers. So I can give more and I can explain more. I'll have you walk through this and give us the lowdown from top to bottom, and we can pull out and pick out specific parts that we really want to dive deep into.
Yes. And just to provide more context, so the Netflix, Varun at True Diagnostic and I were part of the Netflix documentary and were co-leading this epigenetic analysis, which actually was not anyway at the core of the main study published so far and of the Netflix documentary. So we are now really going to talk about something that nobody has been talking about so far. So so let's first talk about the main study in general, and then talk about the epigenetic results in our preprint. So this was a randomized clinical trial including 22 twin pairs. One twin was randomized to a healthy vegan diet and the other twin to a healthy omnivorous diet. Why we called these diets healthy? Because both diets were without or minimizing the intake of processed food. But, and this is important, they were not low carbohydrate diet. We actually encouraged 6 servings of grains and starchy veggies every day. So this is important also to understand. Now, the strength of this study was that we used twins. share not only, of course, the same age, sex, but also very often the same environment. And all these things can influence also on epigenetics, of course, and the analysis of epigenetic clocks. Now, the study also had some limitations. We only had 44 people. It was of short duration, 8 weeks. And a very important limitation that was by design, so it was not a mistake. We didn't design the study to be isocaloric, which means we encourage people to eat to satiety. And so this resulted in differences between the groups. And so the study, many people accused us that they say that this was an unfair comparison because of this, but the study was not designed to be isocaloric. So what happened, the results, the people were assigned to the vegan diet, ate fewer calories, 200 fewer calories per day, and they ate less sugar, more fiber, they lost more weight, and they showed a slower epigenetic aging according to three main epigenetic clocks that we used for this epigenetic analysis. Now, because we know that all these factors, especially calorie restriction, weight loss, and probably even sugar, fiber, can affect epigenetic aging, we can't conclude whether it was the vegan diet itself or all these other factors that were driving the slower epigenetic age. And unfortunately, this was a common misinterpretation of the study that came up over and over in the media coverage. The study was a robust study. The bad part was the misinterpretation and the polarization, because of course the stark contraposition of vegan versus omnivore, and also the Netflix documentary on our study really led to polarized views, with somebody praising the study as a win for veganism and other people just dismissing the study because it was an unfair comparison. But these views don't represent what we found. So I want to emphasize something that was not really discussed even in the documentary, is that vegan diets without proper supplementation, especially of B12, are epigenetically incomplete. So we were talking about epinutrients, right? And B12 is also an essential vitamin. It's not only essential for epigenetic mechanism, but it's an essential vitamin. And it's found in quantities, in bigger quantities, and more bioavailable forms only in animal foods. It's virtually impossible to get enough B12 on a vegan diet. And many vegans know that, right? It's just we need to inform, or also new people that start the diet, that this is a necessity, right? You can thrive on a vegan diet if you supplement. And you can choose a vegan diet for many reasons. Even I have my sisters that are both vegetarians. So there are many for ideological reasons. Anyway, the point is, the study is not saying that vegan diet are epigenetically healthier. And mechanistic studies. So the clocks, the epigenetic clocks, are great and powerful tool to measure these physiological changes, and we're able to track the improvements, the physiological improvements that we see on a vegan diet. But they don't say anything about the mechanisms. And from mechanistic studies, we know even in mechanistic studies mainly mice, but we know the experiment of the agouti mice, the mice that were genetically identical, and yet one was fat and yellow, and the other mouse was lean and brown. And the difference between the two mice was the diet of the mother that was lacking exactly B12, choline, and other methyl donors. And also choline, it's another nutrient that is very difficult to get in sufficient quantity on a vegan diet. It's possible, but it's difficult. Just to give you an idea, you need like 400 to 500 milligram a day, and you get about 100 milligrams for 200 grams of broccoli plus 50 grams of nuts. It's one-fourth. So you need to work on this. I mean, it's possible, but don't just assume that you are getting enough easily. Whereas one egg yolk provides the same quantity, and some meat. You can combine with meat, some eggs. So there are more options for omnivore people. And one last thing, we did also find that people on a vegan diet reported less diet satisfaction. They ate less protein as percent of calories. They lost more muscle mass. So I think these are also challenges to consider in the long term. So being mindful for the people on a vegan diet to eat enough protein and make the diet enjoyable long term, and perhaps engage in some strength training to counteract the possible loss of muscle mass. And finally, one last conclusion. Of course, for any study, we don't have answers for questions that we didn't ask. So we didn't ask the question of comparing this vegan diet to another type of omnivorous diet, perhaps one that is not as rich in starchy vegetables and grains. So we don't know whether in that context an omnivore diet could provide similar benefits or even superior benefits. We just didn't ask that question. So, and this is just because there have been many, over-interpretation and misinterpretation of the study that I feel it's important to discuss here. So thank you for giving me that opportunity.
Oh, of course. And this is really why I was most excited to chat with you today, is to really dive a little bit deeper in this. I think you and me both as scientists, it's our job, it's our duty to explain the study very clearly. And, I think you've done that. I think what the media can do is grab things, pin ideas against one another. They saw vegan versus omnivore, and it just got really, really taken out of context without, the people who are even probably talking about it, I'm not sure that they actually read the study and understood what was done. So it's a very great study. We're just scratching the surface and starting to understand how diet really affects and modulates the epigenome. When I explain the study as well, all of those points you hit were extremely important in looking at some of those epigenetic clocks, which, by the way, were all, I believe, second or third generation as well. I've talked about that as a theme on my podcast, so hopefully those listeners are familiar. And we are starting to understand more and more about how turning on and off genes, like the agouti mouse trial that you brought up in 2003, I believe. I speak with Randy Girdle in one of my previous episodes where we go through kind of that process on what that looks like. So again, everything we're eating, our environment, that nurture, we're actually affecting that gene expression. The point I was going to make, though, is I really like actually looking at the individual. And this is a new topic I've never talked about before on my podcast. We'll dive deep into it on another episode, but kind of those components or epigenetic biomarker proxies, those EBPs that we can go back and measure in each of the diets to see, again, what's really being affected. So there was one protein in particular called the ribonuclease pancreatic protein that actually changes with vegan diets. And that was documented, 10, 15, even 20 years ago in the literature. And we can actually see that protein change in the vegan diet on an epigenetic level as well. So, I think people like to, pick something from a study and run with it. But, as scientists, we're also running these studies to really gather more data that will only really start to create more studies from and more analyses, right? We're leaving with more questions than answers, which I think is always great as a scientist and as the field continues to grow.
Yeah, you beautifully say that. Yes, I think this is a great study, and studies like these really advance the field in ways that are perhaps not, so immediately meaningful for people's life to understand whether a vegan or an omnivore diet is better, which is actually a question that we shouldn't be asking.
Absolutely.
But it's as you said, I'm also very, very I'm fascinated, and I find very promising. So the use of epigenetics for estimating really changes in enzymatic activity and metabolites, because this is really bringing together all the sciences in personalized nutrition, like metabolome, epigenetics, proteomics, and they will basically converge together and have lots of crosstalks that will advance the field.
Of course. Yeah, so we're getting to the end here for everyone listening. Lucia, a couple more questions. What's next for you? Where can people find you? We'll make sure we link everything. You have a couple courses and really fun things that we can give to the listeners as well if they want to learn more. But yeah, tell us what's next. Are you still extending one of these studies? And then where can people find you?
Yes, so I will academically still, I'm at Stanford, I will keep researching epigenetics and diet, and I'm focusing now on education. So education for non-experts and healthcare practitioners. I have a new brand course for non-experts. You can go over at Dr. Aronica, draronica.com, and check out my new course where I've condensed my university teachings in accessible lessons for non-experts. It's perfect for anyone who wants to dive deeper in the topic, whether it's for work or personal interest. And you can also take a sneak peek for free in the first lecture and get a 20% discount if you click in the link in the video description. And you can, I'm also now focusing on my Italian audience. So I have a YouTube channel in English, Lucia Aronica, and I've started one in Italian. I'm writing my first book on epigenetics and diet in Italian. If it's, I will consider to translate it in English as well. And I'm creating some courses for healthcare practitioner. There is already one out with the health optimization medicine, and one coming with the metabolic health initiative probably next year. And I'm happy to partner with new courses. I've been teaching this topic. I established the first nutrigenomic course on epigenetics and nutrition at Stanford for the Stanford Professional Certificate. And so I'm happy to collaborate with other organizations. You can find me on LinkedIn and YouTube at Lucia Aronica, mainly, and on my website at draronica.com.
Yeah. I'll link all of those. Google her name. She'll pop up. This has been amazing, Lucia. I really appreciate your time. I'm going to have to either learn Italian or get the book translated to English, because I'm definitely going to want to read it. So there are so many great assets. I have one final question I ask all of my guests at the end as well. Lucia, if you could be any animal in the world, what would you be and why?
This is difficult.
You've thought about it then, huh?
I don't know,
It's a curveball. It wasn't on the agenda, right?
Yeah. I'm not sure. I think I would be a deep sea animal, just because I very often feel like the ocean myself. Very calm at the bottom, and very excited and bubbly on the surface. This is me. Like
I love it.
yeah. So I think, I don't know which animal, but we with something like a deep water animal.
Something like that. Yeah, I haven't given an answer yet either, but I would go with something I think in the water too, and I don't really know why yet. So cool. We'll leave it at that. Thanks everyone for joining us at the Everything Epigenetics podcast, and remember, you have control over your epigenetics. So tune in next time to learn more. Thanks, Lucia.
Bye.